let's start. People can hear, correct? Anyone?
Yes, sir.
Okay. Okay, let's get started. So today we'll discuss some of the aspects of RNA and CRISPR, RNA to CRISPR-Cas technology. So basically, both of these are also part of non-coding RNA, and these are recent RNAs which have been discovered or in the utilization. Circular RNA is not so long ago which has come in. CRISPR came in only in 2011, I think. So it's very novel types of RNAs which is very much in hot right now in research. Okay, so circular RNAs. If we talk about, we were talking about RNA. So in this case, most of the conventional RNAs are linear, but definitely if we look in the natural surrounding, even in the human cells or as a genome in viruses, many of the RNAs are circular in nature. So generally, if we look into circularization process, again, this has to be a phosphodiester bond formation. And in the general scenario, when we have these nucleotides linkage, it's generally a 5' to 3' phosphodiester bond, correct? So 5' phosphate is linked to 3' OH to generate the phosphodiester bond linkage, and you form the linear polymer of nucleotides which yields into RNAs, and it is the same in the case of DNA. But during circularization, when it has to form a circular structure, it can, in the case of RNAs, because of the hydroxyl groups at the 3' end side, can also have a free OH hydroxyl group at the 2' position, 2' carbon position. So in this case, there are two options for its circularization. It can be 5' to 3' linkage, as you can see, and then very commonly it's 5' OH or phosphate to OH of the 2'. This is very common. So where do we find all these circular RNA? What is the importance of these circular RNA? And circular RNAs are very, very common in the viral genome world or virus world. So that used as a template for the replication to make it more replicative position. So RNA viruses do have circular RNA genomes as well, and it may these circular RNAs may help in the rearrangement of the exons that we are not going to discuss in detail in this case in today's class, because we want to just go ahead with simple understanding of circular RNAs. And these are some of the factors which are being there. And as we all know, because of the extra hydroxyl group at the 2' position in the case of RNA, it makes the double strand formation or the base pairing is not so stable, and it is more prone to hydrolysis that we talk in our DNA or RNA structure studies. So now once you circularize it, because most of the degradation of the DNA or RNA is generally involve lots of exonucleases. We talk about exonucleases as the nucleases which degrades the DNA or RNA from the side ends, from the ends. Now, being circular, the circular RNAs can have doesn't have any ends, so it's not accessible to exonucleases. What does that mean is that circular RNAs are going to be more stable inside the cell or in the environmental setting. So, and again, this circularization also improves because again, the base pairing or secondary structure, always we talk about even the DNA, there is breeding. It's not always double-stranded. There is time separation at the ends of the DNA. So in the case of secondary structure for RNAs, this is going to be much more breeding-wise. It spends more time in breeding in the case of RNA. So now once it's circularized, it gives.
Sir,
yes, tell me.
will endonuclease activity be affected by circularized RNA?
No, that is not. But most of the degradation is done through exonucleases. Most of them,
okay.
most of them, yeah. Until unless it is very specialized, for example, the maturation of the small nuclear RNA and all, you have very specific endonucleases. Generally, most of them are very specific. But general non-specific degradation is done by exonucleases. So it has a lot of contribution from exonucleases. So
okay.
Once you circularize it, it helps in the better, stability of that RNA molecule. And for structurally, just now what I was telling you is that when there is the ends can breed, and then this allows access of the exonucleases to come and degrade better, or it gives you more flexibility. So now once it is circularizes at the ends, that breeding, the structural constraint is going to be very less. The breeding is going to be very, very less. So it is more stable, or the structure which is going to be recognized by certain RNA binding proteins can recognize that structure more often. So it can increases the binding affinity as well. Yeah. So going ahead, as I was telling you just now, that there are 3 or 4 ways wherein you can the RNA circularization can help. One is as a genome or template for replication. RNA polymers can keep on replicating. This gives you multiple copies without even dropping out and then rejoining and finding the origin of replication or start point of the polymerization. So that is one thing. Second is, as I told you, that in many of the places, this circularization helps in getting different sequence of the order of the sequences can be changed. For example, here, ABC elements, if it is opened up at this position, this is going to give you CAB sequence. But if it is clipped here, it is going to give you BCA in the linear format. So this gives you a chance to modify the order of the sequences in the linear format. Another that we just talked about, circularization helps in the stability of RNA by not allowing exonuclease to access the ends of the RNA. And then, of course, structural constraint, thus comes in. Hopefully you people are hearing because power just went off here. Anyone?
Yes, you're audible.
Oh, okay, okay, then it's fine. So now there are different, depending on the RNA origin and type of linkages, there are different types of general types of RNA circles. The one which we are interested very much often is the normal circularization from the mRNA itself. So for example, here, as I told you, circular RNA, you get the virus, circular genomes like hepatitis delta virus. ISDB has a circular genome which is circularizing at 3', 5' that circularization rather than 2', 5' circularization. And then many of the ribozymes or intronic RNAs generally comes as a byproduct of splicing, and these ones can give rise to 3' or 5' circularization just in many of the ribozymes, which is very common in group 2 introns, which are very much important in the maturation that we discussed earlier during the splicing time. And then you do have process intermediate. What does that mean by the circular RNA processing intermediate? Is that it circularizes certain segment of the primary RNA, and the processing enzymes recognize these circular RNAs and process for maturation. We'll discuss a little bit about this kind of RNAs in the later. And then circular non-coding RNAs, like many of the small nucleolar RNAs are circular. For example, RNase P has a circular RNA component, and you can see here the 3', 5' ligation. And then circular RNA splice exons, including the exons when there is a back splicing. Back splicing means the splicing, if you look at it, the first exon joins to the second, and second joins to the third. But in back splicing, what happens is that the second, rather than joining to the third, second, what it does is that fourth goes back and joins with the second. So we'll see that kind of maturation of circular RNAs in the later slides. So you can have various types of circular RNAs in the natural system, and in human system you will see many of these different types of circular RNAs as well. So first is the circular RNA genome. It serves as a template for rolling circle amplification, which gives a single copy can give rise to multiple copies of the same genome in many of the RNA. And I think this one is not so important, we won't discuss. And then as we talk about circular RNAs, that intronic circular RNA from introns, this comes out as a byproduct of splicing. So you have splicing intermediate, then you have the lariat structure during the as a byproduct of splicing, and these lariat structures can just be chopped at the hanging linear fragment, and you get a stable circular intronic RNA. But if it is, there are certain enzymes which would cut at the branching point of the splicing reaction. That's called the debranching step. It can cut here and become linearized. And this linearization helps in the provides accessibility to exonucleases, and it can be degraded faster. So circular RNAs can be much more stable than the linear RNAs, and many of these intronic RNAs have some cellular functions as well. And as we were talking about intermediate, circular RNA intermediate, in some of the archaea, what it has been seen is that for synthesizing the ribosomal RNA, what it does is that it gets a linear transcript, then it circularizes, and further on, it is processed by endonucleases to give rise to the ribosomal ribosomal RNAs here. So basically, these circular RNAs are recognized by the processing enzymes. Most of these are endonucleases, so it can process and matures the 16S rRNA or 23S rRNA in the case of some of the archaea. And then I think in microbiology we discussed circularly permuted genome of I think FAST we talk about. So likewise, this circular RNA in many cases can be obtained by you get a transcript and you circularize it and give rise to this kind of sequence. This kind of sequences, circularizations are important because in many of the organisms, what it does is that the sequences of the tRNA, the preliminary sequence on the genome, is circularly permuted in the genome. So once you circularize it, and what is the, suppose it is the mature sequence of the tRNA, then CAB, it circularizes first and then processed by an endonuclease to give you a mature tRNA sequences. So you can see in many of the algae and as well as archaea, these are very much utilized to get the mature functional tRNAs. So circularization is very, very important in these organisms. So now coming back to the non-coding RNAs in archaea, some of the archaeas, again, the RNase-P, which is endonuclease, is a first generation ribozymes we know of, means RNA with enzymatic activity. Thus, RNase-P are also circular RNA, and some of the SNO RNAs are also circularized in many of the archaea. So coming back to the very much, which is very much prevalent in our system, in eukarya, in human cell lines as well as human tissues, what it has been observed is that you do find what generally happens is that this acceptor, for example, exon 2 acceptor, and here is the donor during the splicing, and arrows are the intronic region. So now this acceptor, rather than accepting the donor sequence from the first exon, it accepts from the donor of the 3' end of the 4 exon. So what happens is that this exon is ligated to or spliced to the exon 2. So the order becomes a circular RNA. So these are very, very common in human genome or in the human system. And what people have found out is that many of these circular RNAs have functional role, and it's kind of quite hot topic right now because people don't understand whether this can be a splice defect. It can be just a defect during the splicing, or does it have any functional role? And there has been evidence that some of the circular RNAs which are found in the system are quite has functional roles because of the fact that some of the transcripts are processed more towards circular RNA rather than the linear form of the mRNA. So this splicing, there is a preference to circularize and has functional roles. And yeah.
Is there any sequence that makes it sort of predisposed to getting circularized, or is this completely random?
It's not random, but the exact rule how it is making more circularized is not known, but there are some observations people have made. For example, we'll discuss in the next slide. What it does is that, what it says is that, of course, this can be a normal effect. But many a times where the circularization is very, very efficient, what it has been seen is that there are repetitive inverted repeats, like ALU1 sequence. Those are very much the inverted repeats. So that enhances base pairing between the flanking exons. Okay, so once it base pairs, it is much more prone to be spliced together. So it enhances the splicing together. But there are some certain studies, what they have shown is that even if you delete this kind of inverted repeats from those sequences also, still it is very efficient to splice. Back splicing is still very efficient. So this is not very essential, but it enhances the efficiency of circularization. So we don't
So it
know
is
what.
not known exactly what causes this to become circularized instead of linear.
Yeah, there is no very straightforward rule. Is not known.
And sir,
Okay.
is it if you go to the previous slide,
Yeah. Hmm.
these so 4 is linking back to 2 here, right?
Yeah. 4 is back splice to exon 2.
Yeah. So, do you also have, like, for the same sequence?
Hmm.
Do you also have transcripts that have 1, 2, 3, 4 normally
Yeah.
parallel
Yeah.
with?
Yes, definitely, it is there. But some of
So this the
the
same sequence can give both products.
yes, same says it's the same transcriptional unit actually. The promoter is the same, everything is same. Just that during the splicing, there is it can be goes into a circular mode or linear mode. Okay, both
Okay.
of them is possible. Yeah.
Okay, sir. Yeah.
So as we were discussing, these repeated inverted repeats may help in enhancing the circularization, but it is not absolutely essential to have these inverted repeats, because if you delete this inverted repeat also, still circularization happens. Another way people have thought about is that maybe there are RNA binding proteins which recognizes certain sequences in the transcript in such a way that it enhances the circularization. So it can be a sequence dependent or RNA binding protein dependent
So,
enhancement or mediation of the circularization. Yeah, tell me.
so that those blue boxes are the exons or introns, by the way?
Exons. Blue ones are the exons and the lines are the introns.
Okay.
Yeah. So yeah, basically in this case you may get introns containing there. Yeah.
Okay.
So, what is the effect of these stuffs? Are there what people have seen is that many a times these circular RNAs, which are coming out of intronic region or containing exons, what it has been shown is that many of the times it has targets for microRNAs. So, now, for example, so in this case, what we are talking about is that uh, cir- circular RNA 7, okay, circular RNA spawns for MIR7, microRNA 7. In this case, it is a circular RNA and it has a lot of target sites for microRNA 7. So when, as we talked about, circular RNAs are going to be more stable. So what it happens is that it can act as a target for microRNA, okay, and it can protect the mRNAs, uh, which are supposed to be degraded through microRNA 7 in this example. So it can act as a sponge for microRNAs, thereby affecting the change in the expression of target mRNAs. Okay, that is one of the hypotheses people have many times, and there are many examples where it has been shown that circular RNAs act as a sponge for microRNAs, thereby affecting or increasing the expression of mRNAs which were supposed to be knocked down or interfered with microRNAs. That is one thing. Second, another way is that many a times these circular RNAs compete for the splicing, and it may generate alternate splicing patterns, because the circularization, if you have this, just like the permutation of the tRNA in the archaea, it can circularize and give rise to different types of RNA products. Okay, splice products can be obtained from these circular RNA. And again, because the intronic RNAs may have binding sites for proteins, or circular RNAs may have binding site for proteins, RNA binding proteins, it can act as a sink for the RNA binding proteins. So if it is a transcriptional regulator which involves the non-coding RNA, suppose, then in that case, circular RNA can act as a sink for those RNA binding proteins, thereby changing the transcriptome of that cell. Okay, so so far what we know, what we have discussed is that circular RNAs can be present as a genome of the RNA. It can be act as an intermediary for during the transcript processing, or it can have its own function like the RNSP, or it can come out normal splicing, and for fifth is that it may come up because of the circularization or back splicing of the mRNA, and they may have different roles to play. So what people are looking at right now is that you look at the transcriptome and find out whether there is any aberrant or enrichment of particular circular RNA in particular tissues, or particular disease state of an individual, so that you can use this as a biomarker for tissue typing, or for diagnosis of certain disease. For example, in a cancer system, if circular RNAs are very much prevalent in this cancer, then it may be used as a diagnostic tool for marker for diagnosing those kind of system. And as you all know, circular RNAs are going to be much more stable. Even if it comes in the circulation, circular RNAs are going to be much more stable in the system. So it may be a better RNA molecule for diagnosis or any tissue typing. Okay, so that is the take home. So then let's not deal too much in this circular RNA. Let's go ahead. So what we are going to talk is next is the CRISPR RNAs, because it has been very hot, and it's still very hot, and there is every day there are new applications or are coming up. So we'll just briefly go to the CRISPR Cas technology. So far, CRISPR-Cas technology has been involved in genome editing. Before that, before the discovery or utilization of CRISPR-Cas technology, two other genome editing tools were developed. The first one was the zinc finger nucleases, and second one is the TALEN. And as we talk in other classes also, zinc finger nucleases engineering was very, very tough, and you have to design a big protein for targeting a particular site. It was very, very difficult and costly. Then came around 2010, I think, 9-10, TALENs came. TALENs is a little bit modular, but still it requires around 20 amino acids to recognize a triplet. So if you have to target a 20 nucleotide site, then you need to design at least few hundred amino acid long protein. So if you have to target in a genome scale, that is not very much scalable. So then it came the CRISPR-Cas technology, as we will discuss here, that CRISPR-Cas technology is very much scalable, and it has been very popular to do genome editing. So basically, to start out, how it was discovered was CRISPR-Cas is more of involvement of protection against fast infection in bacteria. So the initial story starts with the analysis of E. coli genome, I think, in Japan. And what they have observed is that there was a kind of, you have a sequence conservation and then variable region, then repeat of that same sequence. It's a tandem repeats of particular sequence with certain spaces. Okay, so those were the first discovery. People didn't know what was the function, but now what we know is that from these, when the bacteriophage or viruses, bacterial viruses infects a cell, it injects its DNA, so or genomic material. So this genomic material is processed in such a way that it picks up segments of the genome and inserts in the CRISPR array site. Through there are lot many proteins which are involved in this process. Basically, you have a constant region that is called a constant region, and then you have the spacer region, which is variable region, which have been picked up from the viruses, is inserted in a repeat array. So once this is inserted, what it happens is that it allows the transcription of the this repeat array, resulting in long transcript with constant region. Then you have the spacer 1, then constant region, spacer 2, constant region, spacer 3, like this in a tandem array. Then there is another RNA molecule called tracer RNA. Tracer RNA is independent of this array and comes and binds to the constant sequences through base pairing. Once it base pairs, these repeats are matured to give rise to CRISPR RNA complex. It's a 2 RNA molecule complex. Okay, this free RNA or tracer RNA helps in recruiting the one of the protein called Cas9. Okay, Cas protein or CRISPR associated protein. Cas proteins recognize these conjugate fold and forms a ribonucleoprotein complex. This ribonucleoprotein complex can scan through the genome of the invading virus, then viral genome, and through the recognition of a sequence called palindromic adjacent motif PAM sequence is recognized, and this allows the docking of these complex, the pre-RNA complex along with the Cas9, docks there and clips the genome of the invading virus. So basically, the first round of infection allows recruitment of segment of the genome of the virus to the array, and that array matures to give rise to a CRISPR RNA, cRNA, mature cRNA. Through this, with the Cas9 protein, it cleaves the RNA of the invading DNA in the second round of infection. So basically, it gives immunity to the immunity to these viruses. So basically, CRISPR-Cas system has evolved as the immunity in the bacteria and archaea. So if we look into the Cas9 protein as such, if we look into the domain organization, the Streptococcus pyogenes is the one which is most popular right now for genome editing. So if we look into the domain organization, there are few domains. One is the RAFC domain and HNH motif. Okay, so these RAFC and HNH motifs are very, very important, as we'll come to in the next section. And here, L1 and L2, these are the PAM recognizing sequences. So what happens is that the Cas9 along with the guide RNA, when Cas9 alone is there, you have the SNH motif that we talk about. These are all nuclease domain. Basically, it can cleave DNA. Okay, so when there is no CRISPR RNA bound, all these are, more importantly, the PAM region is disordered, so it cannot bind to the PAM sequence properly. Once you have the CRISPR RNA, that is, once it binds, what it happens is that it goes and seeds with the HNS motif, and now the PAM recognition sequence is positioned now. It's get ready for binding to the PAM sequence. So this complex goes and binds to DNA and search along the DNA to find the target. The finding of target, how it is done is that these already ready, the pre-positioned PAM interacting site, when it scans through, it recognizes a very small sequence. For example, in the case of Streptococcus pyogenes, the PAM sequence is NGZ sequence. So whenever they see N is any nucleotide, the NGZ sequence, it will see, okay, there is a PAM sequence. And then once it recognizes, it stores these complex of Cas9 and the guide RNA, we call it guide RNA or cRNA complex. This cRNA complex, it aligns and search whether there is any complementarity in the nearby region. So next to the PAM, if there is a complementary, it invades the strands of the genome. More importantly, this one is the DNA attacking one. So it goes and invades the double strand DNA region, and it forms the R loop expansion, just like we talk about in DNA repair, the R loop in the recombination and all. You have a bubble kind of stuff is there, so that is formed once you have this. So if there is no matching or there is no base complementarity, then it the stall complex goes ahead and tries to find another site which has complementary region. But once you have a complementary region, it invades the strand and unmelts it. Once you melt it, what happens is that we have two domains in the domain organization, RAFC and HNH motif, correct? So now what happens is that the RAFC and HNH motif cleaves on either side of the double strand DNA. RAFC cleaves in one strand and HNH motif cleaves in the other strand. So that's how when you have a complementary region, there is a target site, then it introduces double strand break on the target DNA. So now how does it goes is how the people have started exploiting this system is that, so as we talked just now, that the complex, once you have a complementary region, for SPCas9, if you have 20 nucleotide matching, it can activate the nucleotides and introduce the double strand break on the target DNA. So these double strand break, if you think about, it can be repaired, because when we talk about double strand breaks are the most dangerous breaks in the system, correct? So it has to be repaired by non-homologous end joining method or homology directed repair way. So that we have discussed in our DNA repair classes, correct? So now NHESA is an error prone and trims the ends in a sharp way, inserts or deletes certain nucleotides, and just ligates it. So in that process, few nucleotides or few nucleotides are inserted or few nucleotides are deleted. Okay, so insertions or deletions, what we call together as indels, are introduced in the genomic site. Now, once we delete or insert certain sequences, what it will happen, if we think about the translation, it reads in triplets, correct? So this triplet reading, once it comes, if there is an extra nucleotide, that frame will be changed. So many times this reading, when it goes one by one, because of these insertions, you get stop codon before the original stop codon, or premature stop codons comes in. So that doesn't allow proper expression of the protein in the case of the proper expression of the protein. Now, the question is that because of this premature stop codon, in mammalian systems, generally those RNAs are defective and there is a system to check it. So it degrades the RNA. It's more like knocking out a gene. So you can use CRISPR-Cas technology to knock out a gene of interest using that specific targeting of the CRISPR-Cas technology. Now, this will be
So,
the, yeah.
when the Cas protein is introduced in a mammalian cell, won't it be degraded because the cell will think it is a foreign entity?
No. That foreign entity comes in when it's extracellular, correct? You are talking about immunity, correct? Immune reaction. So
Yeah,
inside
but
the system, inside the cellular system, there is, it cannot discriminate, correct? It cannot discriminate between whether it's a self or not.
okay.
Yeah, it cannot recognize that way inside the system. But if you put in some motif to get it degraded, then it may degrade. But once it goes inside the system, it is part of the system. It doesn't discriminate. Mostly the human self, non-self recognition is outside the cellular system, correct? It recognizes the cellular surface, correct? You have to have antigen presenting cells and all, no? Then only it recognizes whether certain cells has to be killed or degraded, correct? So inside the system, it's not recognized. So it will be as part of its system. Yeah. So now the question here is, now let's go back here. So as I was telling you, the HNH motif and RAV-C motif, these are nucleus which cleaves the one strand opposite to each other. Correct. So if you mutate HNH motif only, then what will happen? It will cleave only one strand. So it can introduce single strand cutting, or those kind of things are called nicks. Okay, in a double-stranded DNA, if there is a single break in one of the strand, that's called nicks. So if you engineer the Cas9 in such a way that RAV-C motif is mutated or HNS motif alone is mutated, then it can introduce nicks in the DNA. These nicks can be utilized very well in developing certain specificities for the Cas9s. Okay, so you can design the Cas9 in such a way that you can introduce single cut or no cutting, because if you mutate both the motifs, HNS motif and RAV-C together, then it's going to bind more stably at this region, but it is not going to cut there. So these properties are very, very important, because here, Cas9, the normal Cas9 that I described just now, is that it will introduce double strand breaks, and it goes into repair in different ways, and you can introduce insertions or deletions, or you can enhance recombination, because if you see here, if there is double strand break, it has to go into recombination mode or NACJ. So certain percentage, when there is no cut, this is not going to be activated, this is not going to be activated. But once you have double cuts, it's going to introduce or activates NACJ as well as SDR. So it increases recombination at a particular site. That's how people have been using in a zinc finger nuclease also. They introduce a particular specific double strand break and enhances NIGJ or recombination, homologous recombination at that site. Because recombination you can utilize for introducing different, like GFP, if you want to introduce. It has to be through more of recombination base. In that case, you introduce double strand break at that particular site where you want to introduce a GFP. Introduce enhances the rate of the recombination, and there is more chance of that GFP to go in rather than random way if you want to introduce. So there is a more than 1,000-fold efficiency enhancement when you have a specific double strand break introduced. So that's why Cas9 or any other targeted nucleases have been very, very useful. Okay, this is the wild type. Now, as I just talked to you, the HNS motif and RUFC motif, if you mutate it, both of them, in such that there is no activity, that Cas9 is called dead Cas9, dCas9. So when there is no cutting, there is no modification on the DNA, but it is going to stay at that site longer because the PAM sequence recognizes PAM binding site is going to bind the PAM sequence, and through complementarity, it is going to stay at that particular genome location longer. So what does that mean is that you can introduce certain things, like if you tag the Cas9 with a fluorescent protein, for example, GFP, you can see the particular genomic locus by using this dCas9. Or you can take these two, or you can introduce transcriptional activators. So then wherever this dCas9 is sitting there, you can enhance the transcription from this site. So you can introduce for seeing a particular site in the genome, you can change the transcriptional activity, or you can put this as we were talking about all these methylation, methyl transferases, DNA methyl transferases was there, or demethylases. If you put in DNA methyl transferases, what will happen? It will methylate the surrounding DNA sequences, and it is going to shut down transcription. Correct? So you can change, you can modify, either you can knock out, or you can increase the transcription, or you can shut down the transcription. You can do all these things through the modified Cas9. So what people have done is, again, if you look at it, you can see if you have these, whatever tagged one, Cas9 is tagged with multiple fluorescent marker, suppose, then you will be able to see the genomic locus. So you can do chromatin imaging as well. So you can check whether the chromosome 2 particular segment comes near to, because we talk about 3D organization of the genome, now it's very popular. In that case, if you want to see whether the chromosome 2 certain segment is coming near to the chromosome 4 and has a functional importance in certain tissues, then you can check through these Cas9 kind of observation tools. And one more important, what it has come out is that when we analyze the genome of different organisms and try to find out proteins which are related to Cas proteins or CRISPR associated proteins, what it has been shown is that people have found out different types of Cas proteins, and among them it can come out that recently is that there are Cas proteins which targets RNA. Okay, so in this case also, just like the Cas9 from Streptococcus pyogenes which targets DNA, what it does is that it goes and finds a target site in the RNA and cut it. Okay, so once it cuts, it will go into degradation, and you can do, just like in RNAi, you can do knockdown of a particular gene expression. And people have been trying to utilize these for knockdown, and there is a lot of uh, scope to develop these tools into application mode. And for example, one, it is very relevant because of the present scenario of COVID-19. Again, here, one of the protein Cas13. Okay, Cas13 is a RNA targeting Cas. Okay, this, what it does is that it goes and binds to the target RNA and cleaves the target RNA. But one more important thing, what it has been shown is that the Cas13 protein, when there is a target RNA, means, for example, it is targeting a particular GAP specific site, suppose. Once it finds a target, it can even cleave nearby RNA as well. Okay, so what does it mean? If the Cas13 finds a target with the guide RNA or cRNA we talk about, then it can again develop there is a conformational change in the Cas13 in such a way that it can even cleave neighboring RNAs. So not only the target RNA, it can cleave the neighboring RNAs. So what does it mean is that you can use this as a diagnostic tool. For example, here, what people have done is that you have an RNA, both the ends are tagged with something. One is the fluorescent marker at one side, and another side is the quencher. So because in a single molecule, fluorescence marker and quencher, when they are together, there is no fluorescence because the quencher is going to be quenching the fluorescence, and there is no signal of fluorescence. Now, once the Cas13 cleaves this sensor molecule, the fluorescent segment is separated from the quencher. So then there will be fluorescence. Okay, so it can be utilized. So what people have done is you do something like this. You have RNA preparation from a COVID-19 patient or something. You get it and you put it this will be the kind of RNA input you will put in. Then you will supply it with the Cas13 and the guide RNA, which is specific to coronavirus 2. Okay, so then what it happens is that it will activate this property, that neighboring RNA cleaving property will be activated. So then what will happen if you put in this kind of a sensor molecule? If there is a COVID-19 RNA, it is going to find the target and activates the cleaving activity and cleaves the RNA, sensor RNA. So now what it does is, as we talked about, it will give out fluorescence. So it can detect the presence of the target sequence, that is the coronavirus sequence, coronavirus RNA sequence in the sample, and gives it out fluorescence. So this can be one of the diagnostics, and I think people are trying to apply these in MIT has just shown this technology, and they say that it's going to be much faster than the normal assays. Like the classical way of diagnostics is RT-PCR, correct? Reverse transcriptase-based PCR. So that takes around turnaround time is very long. And these assays, once it comes, it can come in 1 hour. You can get the result. Okay, so I think that's all for today, and we will continue in the next class with the other topics, so which are not yet covered yet, but we are almost there. I think 1 or 2 classes should be enough to cover everything what we need to cover in the syllabus. Yeah. So any questions?
Sir?
Yeah.
Is it that RT-PCR, why is it so time consuming? Is that because of sample preparation?
Yeah, sample preparation. And then I think, I don't know, technically, if I see, it should not be that time consuming, but it's costly though, very costly. That is the thing. One thing is very costly, because the reagent we get ourselves right now is like 100 reactions. We buy it for 7,000, 8,000. Correct.
Oh.
But that is without profit. Correct. We are just talking about a reagent. But if you have to go in a clinical setting, you have to pay for the manpower, you have to pay for the machine. Correct. And then you have additional RNA isolation. I'm talking about just doing the quantitative PCR. They do quantitative PCR and then determine whether it's positive or negative. Correct. So quantitative PCR itself, 100 reactions goes around 7,000, 8,000. The RNA isolation right now, I think for 50 isolations, we have to pay around 30,000. So it's around 3,000 something. Then that's why they are saying that each assay you need to pay around 7,000-8,000 rupees. That is not very cheap for Indian settings, correct? These kind of things. And transportation, all these things also. It's quite costly in that way. I think time consuming because of the logistics, I think, because everything you have to transfer, correct? For Kerala, it's okay because you are sending it to the RNA virus. I think we have a national facility in North Kerala, so it is faster. But other parts of country, they have to send to Pune. That will take a lot of time, and then you have to isolate and do the assays. It's a very time-consuming process. That's why. Yeah.
Thank you.
Yeah. Any more questions? Anyone? Otherwise, let's stop here for today. Okay. Then see you in the next class.
Thank you, sir.
Yeah.
Thank you, sir.
Yeah. Thank you.